Hi gentlemen, hope you had a good break over this Chinese New Year holiday. For this part of the chapter, I'll keep it short and we will be focusing on how the different factors affect the rate of photosynthesis. So mainly we'll be focusing on three factors: light intensity, carbon dioxide concentration, and temperature. This particular part of the chapter is really important for your practical as well, as we will be looking at different experimental setup to identify the different variables in an experiment. This is important because this will help you in answering planning questions where you are given a situation to conduct an experiment. So please pay attention to this part of the lesson, and we'll also be looking at sources of error, right? How it affects the accuracy and reliability of an experiment, and we'll be looking at how we can improve. Okay, and we'll be looking at how to improve experimental setup after identifying the source of error. So we'll look at that too. Alright, so hang in there as I go through the different factors before we look at experimental setup. I think by now we should be able to define what photosynthesis is and also provide the word equation. As mentioned in class, the chemical equation is for your own knowledge, so you don't have to worry about getting the chemical equation right, but it's very important for you to be able to write the photosynthesis equation correctly with the conditions required. So as we look at this equation, right, we want to see how when carbon dioxide increases, all right, how it will affect the rate of photosynthesis. I know temperature is not in the equation, but why is temperature such an important factor? It's because, all right, photosynthesis is a chemical reaction that involves enzymes, right? In biology, we are always making link across the different chapters, and when temperature is constant, immediately enzymes should come to your mind. So by the end of this video, you must be able to first explain, all right, how each factor affects the rate of this of photosynthesis, and be able to draw the graph to show the relationship. And not just drawing the graph, you must also be able to describe the graph. So this should ring a bell in your head. What kind of questions would come out when we talk about factors affecting the rate of photosynthesis? Describe and explain the graph. Alright, so I'll run through this three graphs as I go through each factor. So let's begin with the effect of light intensity. As seen in this experimental setup, you can see that there's a lamp that's provided in the experimental setup. But how do we vary light intensity? Alright, I'll pause the video over here for you to enter your answer in the question. Alright, I'm trying out something new where I can insert question into the video. So by now you should be able to identify how to vary the light intensity by varying the distance between lamp and plant. Alright, so I'll show you a video on that. And before I proceed on to the video, let us do a little recap on the purpose of using hydrogen carbonate solution. So by now, you should know that as the name suggests, carbonate. So it's a source of carbon dioxide for the plant to make to photosynthesize. Alright, and as you look at this setup, I want you to also able to identify controlled variable. So what are some controlled variable that you can see in the image? Okay, I'll highlight in blue to show you that. Firstly, the intensity of the lamp, all right, the brightness of the lamp, so that when you vary the distance, it helps to vary the light intensity. All right, the type of lamp as well should be kept constant. All right, the type of plant, all right, should be kept constant, and also the temperature should be kept constant with a water bath. All right, now in the lab, we don't use the traditional method of controlling the water bath by monitoring the thermometer. We use a electronic one, electrostatic, sorry, not electrostatic, all right, we use a electronic water bath instead. So the name for it is electronic controlled water bath. So let's take a look at the video to see how this experiment is being conducted. Okay, so how do we measure the rate of the the rate of photosynthesis? We can count the numbers of bubbles released by the plant per minute. So that will give us a rate when we take the number of bubbles over time taken. So I'll just write it down. Note of bubble over time taken will give you the rate of photosynthesis. Alright, let us ponder over this question. Why would you leave the plant for 5 minutes at each light intensity before starting to count the bubble? I would like you to leave a comment also, and then let's look at the answer after this. So what does that mean to allow the plant to adjust to each light intensity? So at the start of each new light intensity, it will bubble a lot, alright, because the rate of photosynthesis has not stabilized at each of the new light intensity. So we do not want to get a false result on the rate of photosynthesis in the first 5 minutes. So we let the plant stabilize and calibrate itself to the new light intensity. So at this point of time, all right, we can see that counting the number of bubble, it's a possible method to count the rate of photosynthesis, but we are making assumption over here that may affect the accuracy of this experiment, that the bubble that we see are all oxygen that is released during photosynthesis, and they are of the same size. So the rate of photosynthesis that we measure may not be that accurate if these two assumptions do not hold. So at this point of time, I would like you to think, instead of just measuring the number of bubbles, what else can we measure? And as we discuss this question, I bring your attention to sources of error. Sources of errors are errors that affect the accuracy and reliability of the experiment, even if you follow each step provided in the instruction manual. So it's not human error, it's not mistakes that we make in the experiment, but the mistakes, the errors that come about because of the use of instrument in the experiment, the way we measure and record data that may affect the accuracy and reliability. Alright, don't worry about calculating the light intensity, but as you can see in this video, as you increase the distance, the light intensity decreases. So what would you be expecting? More bubbles or less bubbles? So size of bubble is a source of error. So how do we overcome the source of error? We can use a gas syringe to collect the volume of oxygen produced, so we can collect quantitative data, right? Numbers, all right, to show the relationship between the varying light intensity and the rate of photosynthesis. Alright, so we have seen in the setup, the following steps are taken. Air bubbles are given off at the cut end of the plant, and some time is allowed for the rate of bubbling to stabilize before counting, as I've already addressed that. The number of bubbles produced will be counted over a given period of time, and this should be repeated to obtain the average rate of number of bubbles provided. So we can find the average rate of photosynthesis, which will increase the reliability of the experiment. Okay, and then it's repeated by moving the lamp closer to the plant or further away at regular intervals. And what is observed, right? The rate of bubbling increases as the distance of the lamp from the plant decreases. So when the plant is very close to the lamp, rate of photosynthesis increases. So this is the conclusion that we can draw from this experiment over here. But the question now remains, right? Can the rate of photosynthesis continue to increase as light intensity increases? What do y'all think? There will be a point of time where it hits the limit, and no amount of light intensity can further increase the rate of photosynthesis. this brings me to my next point on the concept of limiting factor. So what exactly is limiting factor? We pause to ponder about us on a personal level first. Let's pause and think, all right? What is limiting you from achieving your goals? All right, sometimes when we set really high expectation for ourselves, when we have very high goals for ourselves, and when we don't achieve that, it makes us very disappointed and a bit frustrated as well. But sometimes we have to sit down and think, what is actually limiting ourselves? What is actually pulling us back from achieving what we want? So could it be poor time management, poor study habit, or have you been procrastinating before the exams, or a lack of goal that limits you from achieving what you actually desire? So when you overcome your limiting factor, you'll be able to achieve greater heights, am I right? With proper time management, with good study habit, very little procrastination, and a clear defined goal. When you overcome that, right, it no longer limits you, but pushes you to achieve greater height. So if you agree on this concept of limiting factor, I bring your attention back to how limiting factor is defined in biology. So you can see, if I did plot a graph based on the data collected from the experiment just now, we could see that at the start of the experiment, all right, light is a limiting factor. What does that mean? When light is a limiting factor, when you increase the light intensity, all right, the rate of photosynthesis increases as well, as you can see in the first segment over here. But it will reach a point beyond X where no matter how high light intensity increases, photosynthesis remains the same. So beyond X, all right, light is no longer the limiting factor. It could be other factor that is limiting the plant from achieving higher rate of photosynthesis, but at this point, it's definitely not light. So another graph to show you to re-emphasize on the fact that beyond a certain point, all right, something else is limiting. It could be carbon dioxide, it could be water, it could also be temperature. We do not know unless we conduct experiment to change up one of this factor to see how the graph will change. But we are certain that in this experiment, light is no longer the limiting factor. All right, so that is light intensity. We are going to talk about varying temperature, but at this point, you should be able to draw the temperature photosynthesis graph. If you think about it, we covered in chapter 4 of enzymes, all right, how temperature affects the reaction. And the graph that we will obtain in this experiment is the same. All right. So you see, at low temperature, we discussed before, enzymes are inactive. So that would mean that the rate of photosynthesis is low. All right, but at higher temperature, enzyme denatured. So no enzyme means no reaction. All right, so can you recall the shape of the graph? So how can temperature be varied in this instance? It's definitely not the lamp, but the water bath. Another name for this water bath that we use in the lab, thermostatically controlled water bath. So it's an equipment where you can change up the temperature by a matter of clicks of the button. So first thing first, all right, if we don't have the electronic one in the lab, we have to manually control the temperature. So we can start off with a low temperature of 5 degrees Celsius and then measure the number of bubbles produced over a period of time. and then we vary the temperature to 15, 25, 35, 45, 55 degrees Celsius by adding hot water. So that is the old traditional way of regulating the temperature. I want to point out about this. Alright, it's important to also change up the sodium hydrogen carbonate solution used in each temperature to ensure that the volume of CO2 is kept the same for each temperature. So I think without us looking at a video or trying to figure it out, we should know. All right. Okay. Pardon me, in this slide there's a mistake. So we can see that the rate of temperature, all right, how it affects, okay, that the rate of bubbling will increase as the temperature of the setup increases till a certain point, till optimum temperature. Okay, beyond the optimal temperature, photosynthesis will decrease. All right, pardon the mistake in this slide. So this is how the graph would look like. All right, why it starts off slow is because it is less active, as there is less kinetic energy in the particles. So as the temperature increases, more active, because there is a higher rate of collision, okay, between the enzymes and the substrates. And then at its maximum, all right, at the optimum temp, all right, the maximum rate of photosynthesis will be achieved, and then it will eventually slow down as enzymes are denatured. Okay, so temperature is a really short and sweet one, and now we'll look at the effect of the last factor, carbon dioxide concentration. So how do we vary the carbon dioxide concentration? All right, definitely we have to vary based on the concentration of solution used while keeping the rest the same. Let's take a look at the next video to see how it is done in the lab.
A simple way of monitoring the effect of different CO2 levels on photosynthesis is to use a water plant like Canadian pondweed. As the plant photosynthesizes, it produces oxygen, which is released as bubbles into the water. The amount of oxygen produced in a given time is a measure of the rate of photosynthesis.
So you can see the bubble.
A good source of carbon dioxide is sodium hydrogen carbonate solution. The greater the concentration, the greater the amount of carbon dioxide available to the pondweed. To begin with, the plant is immersed in a 0.01 molar solution. A small funnel attached to a capillary tube is placed over the top of the plant to collect the oxygen produced. After a period of equilibration, the solution is drawn through the capillary tube to remove any air. The oxygen produced is then collected for 3 minutes. After this time, the gas bubble is pulled into the capillary tube so that its length can be measured. It's 19 millimeters long. So, with a 0.01 molar hydrogen carbonate solution, the bubble is 19 millimeters.
So in this experiment, we see how far the bubble travels, because the further it travels means that there are more oxygen being produced.
At a concentration of 0.05, the bubble is 39 millimeters. At 0.1 molar, the bubble is 64. As CO2 concentration increases, so does the size of the oxygen bubble. How has increasing the CO2 level affected the rate of photosynthesis?
Alright, so you can see that temperature is kept constant in this experiment. The only thing that changed is the different concentration of sodium hydrogen carbonate solution used. And then instead of measuring just a bubble, we can use a syringe. Alright, to measure the volume of oxygen gas produced instead, which will give you a more accurate results definitely. And the observation, as seen in the video, the rate of bubbling increases as the concentration of solution increases. But the same thing applies over here. Alright, can the amount of oxygen produced increases with more carbon dioxide provided to the plant? Definitely not. There's only so much that the plant can take in. There's only so much carbon dioxide that the plant can take in for photosynthesis. So you can see over here in this experiment, right? If I were to indicate over here, okay, I'll break the graph into two segments. So over here in segment A, alright, CO2 is limiting. Okay, is the limiting factor. As you can, how do I know? Because as carbon dioxide increases, the rate increases. But after beyond the point X, you can see that, right, CO2 is no longer the limiting factor. We can compare between these two setups, one that uses 0.03% of carbon dioxide and the other at 0.13%. So you can see that when carbon dioxide increases, alright, the rate of photosynthesis increases as well. But after a certain point, right, it no longer increases the rate of photosynthesis. As you can see that the graph remains the same beyond this point. So let's go back to review the source of error, which I've already gone through at the start of this lesson. And we were going to discuss how we can improve the experimental setup. Alright, so as mentioned in the first part of the video, that counting the number of bubble produced is not reliable as the size of bubble varies. Alright, some can be bigger than the other, and this does not give us a good data to use to measure the rate of photosynthesis. So how can it be measured instead? Alright, you can see in the recent video, alright, we can use a gas syringe to measure volume of oxygen. We can also use a data logger attached to an oxygen probe, which will tell you the percentage of oxygen produced at a certain point of time. So you can collect the numbers and then plot a graph accordingly. Okay, so just to wrap things up, 3 different factors. You need to be able to first draw the graph, describe the graph, and explain the graph. Alright, you also need to be able to explain the concept of limiting factor when the question is asked. So for light intensity, alright, beyond the point X, right, it's no longer limiting as the rate of photosynthesis did not change. For carbon dioxide, the same thing applies as well. Alright, by increasing the carbon dioxide concentration, it will increase the rate to a certain point. Alright, so if it's able to increase the rate, it is the limiting factor. But if it can't, it's no longer the limiting factor beyond point X. And last but not least, the temperature graph, which we should be familiar by now, since it has already been covered in chapter 4 on temperature. Alright, that marks the end of this lesson. Short and sweet one, I promise, not going to take you more than 30 minutes. I really hope this video is not more than 30 minutes, but spend the 1 week next week during your cohort program. Sit down, look through the video, fill in your notes, answer the checkpoint question before you attempt worksheet 9.2. So just to remind you before you tell me all sorts of reasons about forgetting, alright, by the 10th of Feb, which is Monday, in week 6, alright, I expect 9.3b notes, which is this one, to be filled in and worksheet 9.2 completed. Alright, and then when we resume, we will go through them in class. That's all. I'll see you back in school real soon. Take care.